The EpiQuik CRISPR-Cas9 Assay ELISA Kit provides a fast and convenient way to quantify the CRISPR-associated protein 9 nuclease. Unlike traditional methods requiring electrophoresis and transfer processes, the EpiQuik kit uses an innovative high-throughput colorimetric assay to quantitatively measure Cas9 protein amount, which can be completed in under 3 hours. In this assay, Cas9 proteins in your samples are tightly and stably spotted on microplate wells. The bound Cas9 is then specifically detected with a high-affinity antibody and quantified colorimetrically using a standard plate reader. The kit includes all essential components needed to perform the assay. To begin, prepare the standard curve by diluting the Cas9 standard control with binding buffer to create a range of concentration points. Arrange the required number of well strips in the plate frame. Next, add binding buffer to each well. Load your prepared standards in duplicate to establish the standard curve. Then, load your samples into duplicate wells in the same manner as the standards. Cover the plate with the provided adhesive film. Incubate at 37 degrees Celsius to allow Cas9 binding. During the last few minutes of incubation, prepare Cas9 detection complex solution by diluting the detection antibody, followed by the signal indicator and the enhancer, as specified in the user guide. When incubation is complete, remove the solution and wash the wells with wash buffer. Add the Cas9 detection complex solution to all wells. Then, cover and incubate at room temperature. Following incubation, wash the wells again to remove unbound detection complexes. Add developer solution and monitor as a blue color develops. When the desired intensity is reached, add stop solution. The color will change from blue to yellow, and the plate is ready for measurement. Read the absorbance at the recommended wavelength. Plot the standard curve using the OD values of the standard concentration points and calculate the Cas9 amounts in your samples. With the EpiQuik kit, you'll achieve fast, sensitive, and dependable quantification of CRISPR-associated protein 9. For more information, visit epigentech.com or contact your local distributor.